Reagents contain a template DNA, PCR primers, nucleotides, PCR buffer, and Taq polymerase. The purpose of a Taq polymerase is heat-tolerant bacterium, and it makes new strands of DNA. PCR primers are a short sequence of nucleotides that provides a starting point for DNA synthesis. Usually it's around 20 nucleotides in length, and 2 primers are used in each PCR reaction, and they are designed so they flank the target region, the region that should be copied. Nucleotides provide a starting point for DNA synthesis, and DNA is what the PCR aims to copy. The PCR buffer provides suitable chemical environment for activity of DNA polymerase.
Now we will be explaining what happens in one cycle of PCR. The first step, denaturation, occurs at 96 degrees Celsius. You heat the reaction strongly to separate or denature the DNA strands. This provides a single-stranded template for the next step. Step 2 is annealing, which occurs between 55 and 65 degrees Celsius. You cool the reaction so the primers can bind to their complementary sequences on the single-stranded template DNA. The third step is extension, which occurs at 72 degrees Celsius. You raise the reaction temperature so the Taq polymerase extends the primers, synthesizing new strands of DNA. So here we are explaining amplification. After at least 3 cycles of PCR, there are 8 copies of DNA left. You can see that each cycle produces 2 copies of the DNA from 1 copy.
In vivo is inside the cell, and in vitro is in the thermocycler. In the thermocycler, DNA is separated during heat denaturation, but in vivo, this occurs with an ATP-dependent helicase. In vitro, polymerases come from bacteria or archaea, but in vivo, they are made by the body. In the thermocycler, polymerases have a much shorter half-life. In vitro, some polymerases don't have a proofreading ability. DNA amplification in a thermocycler targets one specific part of the DNA and makes more of that copy. DNA replication in the cell is when the cell replicates the whole piece of double-stranded DNA instead of a small fragment. Gel electrophoresis is important for interpreting PCR results because it allows you to see directly how many copies were produced and if the correct gene is present. The amount of bands representing base pairs shown on the gel shows how long the copy strand of DNA is.